everyone, and once again welcome to Bioline. Today's video is the next video in the microbiology series, okay, and we will discuss about the acid-fast staining here. We have already discussed about the gram staining and the structure and the component of bacterial cell wall. I'll mention the link of those video in the description box so that people can watch that video also. Okay, so let us start with the acid-fast staining. First, we'll study a little bit introductory part of this training procedure. Okay, it was first discovered by Zeal and then it was later modified by the Nielsen. That's why it is also known as Zeal and Nielsen staining technique. Fine, Zeal and Nielsen staining technique is the another name for acid-fast staining. Fine. So this staining is actually a differential staining procedure. Differential staining means a staining which will help us to categorize the organisms into two or more different categories. So here we are going to divide the bacteria in two categories. One is the acid-fast group and the another one is a non-acid-fast group. Okay, so this staining procedure differentiate between the two groups on the basis of the presence of mycolic acid in the bacterial cell wall. Okay, generally the staining procedure is used for those microorganisms or those species which do not bind with the simple stains like the gram stains. Okay, so for that, because they have that kind of cell wall structure or that kind of component in their cell wall, that they need a little bit harsher or a harder staining technique, which is the acid fast technique. Okay, so the best example for this kind of microorganism is the Mycobacterium genus. All the Mycobacterium species belong to this genera. Okay, and they all are stained by the acid fast staining. Fine. So, because these bacteria have a higher lipid content in their cell wall, okay, particularly they have high amount of mycolic acid in their cell wall, and this mycolic acid is actually it is a group of branched chain hydroxylipids. Okay, this mycolic acid it will prevent the entry of the dyes which would like to bind to the cell, right? And that's why these bacteria like Mycobacterium they are unable to be stained by the simple stain. Okay, that's why here harsher treatment or little bit hard stain that is an acid fast stain is used to stain these kind of bacteria. Fine. So in this bacteria, actually what we are doing, we are just this stain actually, we are using these stain to identify specifically the Mycobacterium tuberculosis species, okay, and also the Mycobacterium leprae species. These are the pathogens or they are the human pathogen and responsible for causing the tuberculosis and leprosy in humans, right? So what are the three components? We can say the four major components are there of this training procedure. The first we are going to use here is a component known as primary stain. Okay, and in this case the primary stain is a carboyl fusion. This carboyl fusion is a mixture of basic fusion and phenol. Okay, apart from that the water is utilized for their mixing process and all, but these are the two basic component of primary stain, carboyl fusion. Okay, apart from that this heat here we will be using heat as a mordant in this staining technique. So what is a mordant? We have already discussed in the previous video. Okay, mordant is an agent which will actually increase the binding affinity of the primary stain to the cell wall, right? It will actually increase the efficiency of this primary stain so that it can interact with the cell wall and the cell wall will get more stain with the primary stain. Okay, so here heat is going to act like a mordant. After that, we'll be using a decolorizing agent or a decolorizer here. That is a 3% HCl in 95% alcohol, or we can say this is a acid alcohol mixture, or we can just simply say this is an acid alcohol decolorizer that we will be using in this staining technique. Lastly, we'll be using another dye or another stain which is known as a counter stain, and in this specific staining purpose we'll be using methylene blue as the counter stain, right? First of all, just we'll quickly go towards the procedure of the staining technique. Okay, first of all we will just take the sample of the bacteria on our slide. Okay, and then we're going to add the carboyl fusion, that is our primary stain, to the slide. Okay, after that, what we will do, we will just apply the heat to the slide so that this carboyl fusion can actually enter, or we can say this primary stain can actually interact more efficiently with the cell wall of the bacteria. Right, after that, definitely there is a washing step so that the excess primary stain may get removed from the slide. Okay, and then we are going to add the decolorizer here, which is the acid alcohol. This decolorizer, what it will do, it will try to remove this primary stain from the bacteria or from our slide. Okay, then again we have to give a washing step here, and later on we are going to add our counter stain, that is our methylene blue. Here we will be adding as a counter stain. Okay, then again we are going to do a washing step so that the excess stain may be removed. We are going to dry the slide and then we can observe it under the oil immersion microscope. Right? So this is the general procedure, stepwise procedure of the acid fast staining. Okay, so let us understand the principle of the acid fast staining here. Okay, now see, first of all, in the very first step, what we are doing after the applying, or after making a smear of the bacteria on our slide, we are applying the very first stain, that is the primary stain, carboyl fusion stain. Okay, once we apply that stain, what the stain will do, it will try to solubilize the lipoidal portion of the Mycobacterium cell wall. Okay, it will solubilize the lipids present in the Mycobacterium. Okay, because I have already told you the lipid percentage of Mycobacteria is higher. Okay, and there is almost very little peptidoglycan present in the Mycobacterial cell wall. Okay, lipid percentage is very high. So, and also why? Because it also has the mycolic acid specifically. Right, mycolic acid is there, so it is going to give the bacteria a waxy appearance. So carboyl fusion can actually solubilize these lipoidal portion. Okay, and then the heat, which is actually the mordant here, this aids the further penetration of this carboyl fusion into the lipoidal wall, and thus the primary stain enters into the cytoplasm of the bacteria. Fine. If this is the bacteria and if this is the cell wall, this is the plasma membrane. Once the heat is given, you can see the primary stain can now easily, it will enter into the cytoplasm of the bacteria and it will stain the cell. Okay, and after this we can see what is happening. You can see this is the heat step, this is the primary stain cell. So after this you can see all the cells will appear reddish in color, which is the color of the carboyl fusion. Right? Okay. Now, in the next step, that is our decolorization step. We are using our acid alcohol for decolorizing. Okay. Now, what happened? When we will use the acid alcohol here, again the mycobacteria, the mycobacteria, what it will do, because again it is having a higher lipoidal content in its cell wall, it will resist the decolorizing effect of the decolorizer. Okay, it will resist the release of these primary stain from the cytoplasm. It will not allow that stain to move out of the cytoplasm. Okay, so even after applying the decolorizing agent, this acid fast category, that is of the mycobacteria, that will not get decolorized and it will still appear reddish in color, which you can see here. Okay, so these are the mycobacteria which are still reddish in color even after addition of the decolorizing agent. Okay, and the other one, okay, and the other cells which may be here like that. Okay, these other cells which were earlier pink in color or reddish in color, okay, because they do not have that mycolic acid in their cell wall and the lipid portion will be lesser, they'll be having more peptidoglycan. So under that condition, under those conditions, this colorizing agent will be able to release the uh, carboyl fusion stain from the cell, and these will appear colorless after the decolorizing step. Okay, we can see here. Okay, they in the non, and that category will be known as non-acid fast. So non-acid fast organism which lack the lipoidal material in their cell wall, due to which they will be easily decolorized and they will appear colorless. Okay, and the Mycobacteria type species, which are also known as the acid fast stain, okay, they are resistant due to the presence of large amount of lipoidal material in their cell wall, and this will prevent the penetration of the decolorizing solution. That's why even after the decolorizing solution they are reddish in color, but the other cells which were earlier reddish, after decolorizing they will become colorless. Okay, now we'll move to the next step. So the next step is just we will counter stain the smear. After, obviously after decolorization we will give a washing step to remove the excess alcohol. Okay, so now we'll add the counter stain, which is a methylene blue. Okay, now see, we saw that the earlier this was the non-acid fasting. Okay, we saw that after decolorizer, after the we can say the addition of the decolorizer, the acid fast bacteria like our Mycobacterium, they're still in reddish in color. That's why when we will apply methylene blue here, they will not take up that stain. Okay, and even after that you can see they will appear reddish in color even after adding the methylene blue. Okay, but the other category, which was the non-acid fast category, which was the colorless cell, because after the decolorization the carbonyl fusion has left the cell and the cells have become colorless. So what will happen now? They are free to take the methylene blue. Okay, so they will take up the methylene blue, which you can see like that. Okay, and they will appear bluish in color, and that category will be the non-acid fast. Okay, that's what is mentioned here, that only the decolorized cell absorb the counter strain and take its color, and they will appear blue, and that category will be non-acid fast, which you can see here. Okay, and the acid fast cells, which were actually red in color, which retain their red color even after decolorization, because why did they retain the color? Because of the presence of excess lipoidal material like mycolic acid. Okay, now they will maintain their red color, they will not take up the methylene. Okay, so that's why this mycolic acid appears to be responsible for this acid fastness. Acid fastness means appearance of the reddish in color. They can still retain the color of the primary state. So this acid fastness characteristic of the acid fast cell is because of the presence of mycolic acid. Okay, so after this we are going to give a wash to the slide, and then again we will see it under the microscope. So this is how we can see if we have a mixed culture. So this is how we can see in the microscope. You can see the reddish one. Okay, the reddish one which can appear from bright red to intensive purple, this B category. These are the reddish one, and these are the acid fast cells. Okay, they have retained the primary state throughout their procedure. Okay, the second category which you can see, which appears bluish in color, because they have taken the color of the methylene blue, the counter stain, this A category. These are the non-acid fast. These are the non-acid fast. Okay, so this is a mixed culture, and this is how we get the result. If you have taken only one mycobacteria, definitely we will only see the bright red or the purplish color. And if you have taken only the other category, okay, like E. coli, for example, you have taken. So E. coli is a non-acid fast stain, non-acid fast bacteria, and it will appear bluish in color. Right. Lastly, we'll just summarize this whole staining. Okay, you can see here these are the reagent, the application of the dyes, and the cell color. Okay, after the addition of the primary dye, which is the carboyl fusion, both of the categories, the acid fast cell and the non-acid fast cell, they will appear reddish and reddish in color. Okay, primary dye plus we are giving heat here so that the carboyl fusion can enter or penetrate into the cell wall and the plasma membrane. Okay, and this carboyl fusion will devoid the mycolic acid or the lipoidal content. It will try to dissolve those materials. Further, after the addition of the decolorizer, which is the acid alcohol, the acid fast stain will retain the red color, but the non-acid fast stain will become colorless. Why they will retain? Just now we have talked about this, that the mycolic acid or the lipid content will not allow the primary stain which is now present within the cytoplasm of the cell to leave the cell. Okay, it will resist the primary stain to leave the cell. Okay, lastly we have the counter strain, that is methylene blue. Definitely, because these acid fast cells, they are already reddish in color, so they will not take up the the counter stain, and they will appear reddish in color. And the non-acid fast category, which is colorless, after addition of the decolorizer, they can easily take up the counter stain, which is the methylene. Okay, so that's all for this staining procedure. Okay, I'll mention the links of the extra videos in the description box. You people can watch those videos, okay, and relate to this staining procedure. Okay, we'll meet in the next video. Till then, have a good day.